Hi guys, welcome to another section of Learning with Prepclass. I am Tutor Chima, and today we are going to be looking at Jamb Biology 2017. Now, in today's section, we are going to be solving questions 6 to 10 because in the previous video we looked at questions 1 to 5. So today we are going to be looking at questions 6 to 10 of Jamb Biology 2017. Alright guys, before we proceed, do not forget to subscribe to our YouTube channel, like our videos. When you look below there, you are going to see a notification bell. I advise you to click the notification bell and put it on. Once your notification bell has been activated, you are going to be the first to be notified whenever you have an educational content like this on our channel. Always feel free to drop 1 or 2 questions on our comments section box. Question on this video, question on the previous video, and we'll be available to answer your questions. Alright guys, you can also be kind enough to share our videos so that it's going to reach as much viewers as possible. So without wasting your time, let us go straight to business. Question number 6. The organelle responsible for osmoregulation in Paramecium is? The organelle responsible for osmoregulation in Paramecium is? Is it a flame cell? Is it the nephridia? Is it a contractile vacuole? Is it a mammalian tubules? Now, all these organs listed here are osmoregulatory organs of some organisms. Flame cell, in flatworms, nephridia, in earthworm, mammalian tubules, in insects. So they're not asking us which of them is the osmoregulatory organ for Paramecium. I believe you know what's osmoregulation. The process whereby an organism balances the level of water and solutes in its cell. Like in humans, our osmoregulatory organ is a kidney. We use the kidney to conserve water, to balance the level of water and salt or solute in our blood. So now, the contractile vacuole is responsible for removing excess water that enters into the cells of unicellular organisms. In this way, it is responsible for osmoregulation. You know, these unicellular organisms like amoeba, Paramecium, euglena, chlamydomonas, they are mostly found in fresh water habitats. And because they are found in fresh water habitat and because their cell is very, very thin, water can easily permeate into the cell. So water can easily enter into the cell and dilute the cell. It can cause the cell to swell and rupture or burst. Now, for the cell, for that unicellular organism or that particular cell to be alive, it must have an organelle that helps it to remove excess water that enters into the cell in order to prevent it from swelling and bursting in the case of an animal cell. And that osmoregulatory organ is the contractile vacuole. As water enters the cell, the contractile vacuole becomes filled and goes to a corner of the cell membrane and release that excess water out into the hot environment. So the contractile vacuole is responsible or is the osmoregulatory organ for unicellular organisms like amoeba, Paramecium, euglena, and so on and so forth. So the answer is contractile vacuole. 7. The excretory organ of an earthworm is represented by, I believe in the last question I must have mentioned that the excretory organ of an earthworm is represented by, is it 4? Is it 1? Is it 3? Is it 2? Is it 4? Diagram 4. Is it diagram 1? Is it diagram 3? Is it diagram 2? What are these diagrams? Diagram 1 is a flame cell. This one here is a flame cell and it's the excretory organ of flatworms like tapeworms, planaria. Diagram 2 is the kidney and is the excretory organ of mammals, humans, monkeys, dogs, cats. Diagram 3 is the nephridium. Nephridium singular, nephridia plural. It is the osmoregulatory or the excretory organ of earthworms. Diagram 4 is the mammalian tubules and it is the excretory organ of insects. So you can see that the answer here is what? 3, which is option C. So 3 is the osmoregulatory organ of earthworm and it's called the nephridium. So the answer is 3. 3. 8. The platelets in mammalian blood are responsible for, is it transporting oxygen? Is it initiating clotting? Is it removing carbon dioxide? Is it destroying microorganisms? Now, we have 3 blood cells. You have the white blood cells, you have the red blood cells, you have the platelets. The white blood cells are responsible for fighting and killing germs. The red blood cells are responsible for transporting oxygen, while the platelets are responsible for blood clotting. Once a tissue is broken, the platelets are responsible for clothing and covering that tissue. I think whereby thrombin is converted to prothrombin converted to thrombin, that now causes another protein, fibrinogen, to be converted to threads of fibrin that now entangles the cell and forms a cloth. And that is the job of the platelets. So the platelets are responsible for blood clotting. A good or well-clotted, a proper blood clotting helps to prevent hemorrhage. Hemorrhage simply means loss of blood. So that's the essence of the platelets, to prevent what? Loss of blood. So the answer is initiate blood clotting. That's the job of the platelets. 9. The most important factor that determines the different types of vegetation is it light? Is it wind? Is it temperature? Is it rainfall? Now, they didn't ask this question maybe since they are mentioning vegetation, they are talking about terrestrial habitat. So the most important factor that determines the different types of vegetation in terrestrial habitat is vegetation simply means plants cover. Types of plants that grows in a particular area, trees, grasses, sedges, are you following? And what is responsible for that type of plant that grows in a particular area is rainfall. Rainfall is the most important. The higher the rainfall, the denser the plant population in that particular area. So the amount of rainfall determines the type of plants that grow in a particular area, or the type of vegetation seen in a particular terrestrial habitat. Just like in the southern part of Nigeria, we mostly have the rainforest zone because rain falls so much in that area. Rain can fall as much as, even as high as 9 months in a year. You go to places like the riverine, like River State, the Bayelsa, places like Lagos, rain can fall about 6 to 8 months in a year. So those kind of areas usually have what we call tropical rainforest, very dense forest, full of trees and grasses. When you move to the middle belt, you might see the savanna area, sparse vegetation and trees. When you now move towards the north, you see the savanna, and that's why the northern part of Nigeria are mostly the savanna, and they are mostly desert-like because they usually have a very low amount of rainfall. So that's why trees are usually scarce in those areas. We cannot go up north now, like in let's say you go to Sokoto, or you go to an area like Yobe in Nigeria, and you say you want to start growing plantain, or you want to start planting tree crops like oranges and the rest of them. They may not do very well there. Why? Because rainfall is very low. So rainfall usually affects the kind of vegetation cover in a particular terrestrial habitat. So the answer is rainfall. 10. When testing for the presence of starch in a leaf, the reason for dropping the decolorized leaf in hot water is it to detect the starch? Is it to kill the leaf? Is it to soften the leaf? Is it to remove the chlorophyll? Now, I don't know if you've performed this experiment when you are in school, where I want to use a leaf that has been photosynthesizing to test for the presence of starch. That leaf is first of all harvested, then it's dropped in boiling water for some seconds, so about a minute or so, to kill the leaf. Boiling water first to kill the leaf. After that, we remove the leaf. We now place it inside hot ethanol or alcohol. The hot ethanol alcohol removes the chlorophyll. Are you following? After it has removed the chlorophyll, the leaf becomes very tough or strong. Very strong, just stand and become very firm. Then we now drop the leaf again in hot water. That hot water, after the ethanol or the alcohol has removed the chlorophyll, the leaf will become pale, almost white in color, but very tough. You now place that leaf in hot water. That hot water this time around is to soften the leaf, is to make it soft. After the leaf has been decolorized, that hot water now helps to make the leaf to become soft. So I said that the hot water helps to soften the leaf after decolorization with hot alcohol. The leaf becomes the hot water helps to soften the leaf, yes. Now after decolorization with hot alcohol, the leaf becomes hard or tough, just like as I explained. Now the hot water makes it soft before addition of iodine to it. So once the leaf has moved for that alcohol, hot alcohol that removes all the green color or the chlorophyll, making it very strong and tough, you now drop it inside hot water to soften it. After that, you place it on a white tile and you drop 3 to 4 drops of iodine solution to it. Once you do that, if that leaf has been actively photosynthesizing, it's going to turn what? Purple in color, or dark purple, something dark purple in color, as indigo, that kind of purple, dark deep purple, or almost black in color, telling you that that leaf has been actively photosynthesizing. So the correct answer there is to soften the leaf. That's the function of the hot water, to soften the leaf. Alright guys, we've come to the end of today's section of Learning with Prepclass, where we looked at past questions on biology 2017 Jamb. We solved questions 6 to 10. Now if you look here, you see a link. I urge you to type this link on your browser and read it. When you do so, it's going to take you straight to our WhatsApp group. When you join us on WhatsApp, you're getting updates, you're getting infos on the latest content we have on our channel. Alright, till you see my video again, I still remain Tochima. Bye, and do stay safe.